Okay, so this is part 2 of our recombinant DNA and biotechnology lecture. And this one is going to concentrate on how we can use restriction endonucleases to manipulate what genes are present in an organism to actually move genes from one organism to another. And again, we talked about this in the first lecture. The basic premise of recombinant DNA is to cut 2 different samples of DNA from 2 different organisms with the same restriction enzyme that creates pieces of DNA with complementary tails with sticky ends. And if we combine those and add some ligase to join them together, we can get a piece of DNA that is recombined. That is, it consists of DNA from 2 different organisms. So we can use this technique to take a gene from one organism and insert that into the genome of another organism. And that's the basics of recombinant DNA technology. Now, this was first done using plasmids, bacterial plasmids, as a vector, as the means of transferring genes from one organism to another. So plasmids are small circular pieces of DNA, often described as extrachromosomal DNA. So the larger piece of circular DNA in red in this diagram is the bacterial chromosome or the bacterial DNA. These smaller circles are plasmids. They tend to be a few thousand base pairs in size. They can have particular genes that are located on them. They're easy to isolate from bacteria. So once they're isolated, then we can expose them to a restriction endonuclease and cut them and leave sticky ends that allow us to insert other genes into the plasmids, and then we just reinsert those genes back into a bacterial cell. So that's the basic premise of doing recombinant DNA. So in the upper left, we can see a bacterial cell with a plasmid. We're going to isolate the plasmid, remove it from the bacterial cell, cut it with a restriction enzyme. We're going to take some DNA that we want to insert and cut that with the same restriction endonuclease. So we get a piece of DNA that has sticky ends that's complementary to the sticky ends that were produced in the plasmid. We simply combine them and add some ligase so that they'll join together. And we now have a recombined plasmid, a plasmid that has the introduced gene from our source. And we simply introduce that plasmid back into a bacterial cell. And now that bacterial cell has that plasmid. And if it can transcribe and translate that gene, it will produce whatever protein product was coded for by the piece of DNA that we inserted. So we can essentially induce a bacterial cell to produce the product of any gene that we can isolate with a restriction endonuclease. Now, it sounds easy. You got to do some manipulation to make sure that this works properly, and you can identify the bacterial cell that have been recombined. So one thing that is done is to only use plasmids that have genes for resistance to an antibiotic within the genome of the plasmid. So this is a particular plasmid that's found in E. coli. It's called PUC18, and it has an ampicillin-resistant gene, meaning that any bacterial cell that has this plasmid would be resistant to the antibiotic ampicillin. That is, it would grow in any medium that contains ampicillin. And any bacterial cell that doesn't have this plasmid would be killed by the ampicillin. It wouldn't survive. So this is a way in which we can identify cells that have a plasmid, have properly taken up the plasmid. And the other thing we want to do is manipulate it so we can identify those cells that have plasmids that have had recombinant DNA inserted. And we do this by looking at a particular gene in that plasmid, the LAC-Z gene. This is the one that's responsible for producing the enzyme that breaks down lactose. So any cell that has this gene would be able to survive if lactose is provided in the medium on the Petri dish. And in that particular gene, there are restriction sites for a whole number of different restriction endonucleases, which you can see listed in the upper right, including EcoR1. So we could expose this particular plasmid to EcoR1. It would open up the plasmid at that site, and then we could insert a gene in the plasmid at that site. And what that would do is disrupt the LAC-Z gene. The LAC-Z gene would no longer be able to code for the enzyme to break down lactose because it would have a whole bunch of base pairs, another gene inserted into the middle of that gene that would render the LAC-Z gene non-functional. So any cell that has a recombined plasmid would not be able to use lactose as an energy source. So it's a way in which we can isolate those cells that not only have plasmids because they're ampicillin resistant, but we can also identify them as being recombined, having recombined plasmids, because the LAC-Z gene, the LAC-Z enzyme would not be produced. And we can distinguish those cells and isolate them. Then we know that those cells have been recombined. So again, the process involves isolating the plasmid with ampicillin resistant, cutting the DNA with a restriction enzyme in the middle of the LAC-Z gene, inserting the DNA that we want to insert into this cell by cutting that DNA with the same restriction endonuclease so that it has sticky ends. Those sticky ends will bind to the sticky ends of the plasmid, and our gene has been inserted. And then we simply put that plasmid back into a host cell. We identified, because that cell is ampicillin resistant and cannot use lactose as an energy source. And the way that this system works is that if they can use lactose as an energy source, they form a blue colony of cells. If they cannot, they form a white colony of cells. And so we can easily distinguish the colonies of bacteria that have been transformed from those that have not. So you can easily distinguish between the blue colonies and the white colonies in this picture. Okay, so the next question then I think becomes, where do we get the DNA that we want to insert? And so if we want to insert a human gene for a particular protein product into a bacterial cell, into a plasmid, and insert that into a bacterial cell, we have to identify the right gene, the piece of DNA that has the right gene. And that's the challenge. One technique is to create what's called a genomic library. So you take a sample of human DNA, you cut out the restriction enzyme, so you have lots of different DNA fragments. You combine those with plasmids and insert them into bacterial cells, and you're going to get hundreds or maybe thousands of different strains of bacteria, each which have plasmids with different pieces of DNA in them. And that's a genomic library. It's essentially a set of cells that have different pieces of human DNA inserted in them. And then we need to screen all of those bacterial cells to try to identify those that have the gene of interest. And so what we do is take each one of those and grow them out into colonies on a petri dish, transfer the DNA to a piece of filter paper, kind of like what we did in the southern blot, the DNA fingerprinting technique. And then we create a sample of probe in which small pieces of DNA that are radioactively labeled, that are complementary to sequences of DNA in the gene that we're trying to identify. So we expose the piece of filter paper to that probe. If the probe finds that particular sequence of DNA that it's complementary to, it will bind to it. And because it carries that piece of radioactivity, we can identify that colony, and we can isolate that colony and grow it in culture. And now we have a colony that we know has the gene of interest because it has DNA that is complementary to our probe. And that probe was constructed from our gene of interest. So if, for instance, if we're looking to be able to put the insulin gene, the gene for the protein, human protein insulin, into a bacterial cell, we would create a probe based on the sequence of DNA and insulin, use this technique to identify those colonies that have plasmids that have that piece of DNA, human DNA inserted. And once we have, then we can simply allow those cells to proliferate, and we can manipulate those cells so they'll transcribe and translate that human gene and produce insulin. Another technique for getting a particular piece of DNA is to look for a piece of messenger RNA for the gene that we want to encode. So you go to a cell that normally transcribes that piece of DNA. So again, if we're looking for insulin, we would go to the cells in the pancreas that make insulin, and we look in their cytoplasm when those cells are active and functioning, and isolate the mRNA. And some of that mRNA would be mRNA for the insulin gene. And what we do is use that piece of mRNA to make a double-stranded piece of DNA whose sequence of bases is based on the sequence of the mRNA. So we start with that piece of mRNA that we've isolated from the pancreas cell. We add a small piece of primer to it, and then we allow transcription to occur, so that or reverse transcription to occur, so that a piece of single-stranded DNA is produced using the piece of RNA as a template. And then we remove the RNA, add some DNA polymerase, and allow the second strand of DNA to be produced, add a little ligase to join the pieces together. And now we have a piece of double-stranded DNA that's called cDNA or copy DNA that has a sequence that is based on the sequence of bases of the mRNA. And presumably that mRNA is the mRNA that's used to produce insulin. So now we have a piece of DNA that has that insulin gene, that insulin sequence, and we can insert this piece of DNA into a plasmid and put it into a bacterial cell, so it can be induced to make insulin rather than other kinds of bacterial proteins. So insulin is actually a little bit more complicated than that. Insulin is transcribed from two different genes, and there are two different polypeptide chains that make up a functioning insulin molecule. There's chain A and chain B. And they're produced from the same pre-mRNA, but in both cases, after translation occurs, a bunch of amino acids are removed between the A chain and the B chain to produce the finished product. And so what you need to do is produce both of these, the B chain and the A chain. And so what you can do, again, is isolate the DNA for the B chain or isolate the DNA for the A chain, insert that into a plasmid, and that's what's shown here. Inserting that into a plasmid, allow the plasmid to go ahead and make that particular chain of amino acids. And then once we produce the beta chain, we can combine it with the alpha chain, and we now have insulin. And so this is the technique that is now used to produce the insulin that diabetics use. Prior to these genetic engineering techniques, insulin was acquired from the pancreases of many different animals like horses and cows and pigs and sheep. And that insulin is close enough to human insulin that it would be functional. And so they would identify, extract the pancreas of each of those animals when they're slaughtered, isolate the insulin that's being produced by those cells and purify it and concentrate it, and use that to make the insulin that a person who has diabetes would use. That caused all sorts of problems. The DNA or the protein wasn't always properly purified, and so other proteins might be included, and that could lead to the development of allergic reactions, depending on the source of the insulin, and it eventually caused problems for those patients. So this is a much better technique. It's actually producing human insulin that is identical in sequence of amino acid to the insulin that everyone produces. And it doesn't have any of the contamination that might occur by trying to extract insulin and purify it from another animal. So it's a great technique now to produce a drug that people with diabetes have to have if they're going to stay alive. And it turns out dozens, maybe even hundreds of different kinds of proteins that humans might need to treat particular diseases. They've been identified and techniques have been developed in order to induce bacterial cells to produce them. And so here's a short list of them. Insulin is at the top, and again, it's used to treat diabetes, but human growth hormone can be produced to treat individuals that have pituitary dwarfism. Blood clotting factors can be produced for people that have hemophilia. Various kinds of enzymes that are necessary to treat someone with cystic fibrosis. All sorts of diseases now can be treated by using this technology. So this is one very important and very useful outcome of the whole biotechnology industry, is to produce proteins that can be used to treat various kinds of human diseases. Now, one problem with these techniques is that there's size limitation. Plasmids generally are only a few thousand base pairs, and it's possible to use a plasmid as a vector to insert a gene into a bacterial cell only if the plasmid is up to about 20,000 base pairs in size. And there are many genes for human proteins that we'd like to be able to produce that are much bigger than that. So we have to use something else other than a plasmid. And one technique is to use yeast cells as the vector and to create what's called a yeast artificial chromosome, or a YAC, Y-A-C. And so this is an example of the yeast artificial chromosome. It has to be specially constructed. It's got to include an origin of replication. It has to include the sequence for the centromere. It's got to include sequences for telomeres. But by manipulating it with restriction endonucleases, we can insert a particular gene into this artificial chromosome and then insert that chromosome into a yeast cell. And now we can induce a yeast cell to produce the protein that's coded for by a human gene. And again, the benefit is that we can insert much larger genes, genes that are perhaps bigger than 1,000 kilobases, 100 kilobases, rather than the 20 kilobases that we're restricted to in plasmids. So this is an important technique for being able to produce the products of large human genes, larger than we can use with a plasmid. There are techniques that can be used to insert genes in plants. There actually is a naturally occurring bacteria that's called agrobacterium that will insert a plasmid into a plant cell. And they do this for a purpose that's beneficial to the bacteria. When they do that, they're inserting genes that disrupt the normal growth of the plant to create what's called a gall. And that's what's in this bottom picture. It's essentially a huge plant tumor. And this plant tumor supports the bacteria. It's a place where the bacteria live. And so bacteria are doing this in order to manipulate the growth of the plant cell in ways that are beneficial to the bacteria. So we can take advantage of this. And we do this by putting particular genes into that plasmid, inserting them into the bacteria cell, allowing the bacteria cell then to insert the plasmid into the plant cell. And now that plant cell is going to be transcribing and translating the genes that we have inserted into the plasmid. So it's a real handy technique for inserting certain kinds of genes and producing them in certain kinds of plants. And so this slide kind of summarizes that process. You isolate the plasmid, insert the gene in the plasmid, insert that plasmid then into the bacterial cell, allow the bacterial cell to insert the plasmid into a plant cell, and then you manipulate that plant cell in culture. And you grow it in culture, provide various kinds of growth modifiers that will trigger it to grow into a plant, a transgenic plant, and then you can put that plant in the ground, and we can now use plants then to produce all sorts of different kinds of proteins, essentially proteins that might be used to treat particular human diseases. Here's one example of that. I won't go into a lot of detail of the genetics of it, but essentially what we're trying to do is isolate the genes that are necessary to produce beta carotene. And beta carotene is an important source of vitamins. Carrots are loaded with beta carotene. You've probably all heard that you should eat lots of carrots in order to have good night vision, because the beta carotene is involved in vitamin A metabolism. And so vitamin A is necessary for night vision. So eating lots of carrots gives you lots of beta carotene that promotes night vision. But there are people who live in parts of the world where the principal source of food is rice, and they don't produce any carrots. So rice does not naturally produce beta carotene. Those individuals are much more susceptible to illnesses of the eye, especially problems with night vision and things. So rather than just give them beta carotene as a vitamin or something, why not manipulate the food that they normally eat to produce a crop that includes the genes necessary in order to make beta carotene? And so that's what's been done, and it produces a strain of rice, a transgenic strain of rice that can now produce beta carotene. So on the picture on the bottom, you've got the white rice in the lower right. It doesn't produce beta carotene, and the one on the left does. So just by inserting this particular gene using these techniques, we can produce a much more nutritional form of rice that can solve a vitamin deficiency for people that live in certain parts of the world. We can even manipulate animals. There are techniques that we can insert particular genes, like a growth hormone gene, into the genome of an egg cell, a fertilized egg cell, and it's associated with a particular promoter. And once that promoter initiates transcription, then this growth hormone gene will produce its product, which is to enhance the growth rate of an animal. And so in this case, we can produce salmon where the only difference between them and the wild type is the existence of this particular growth hormone gene. We can induce them to grow much more rapidly and really ramp up the food production. And as the world population grows, exceeding 7.5 billion now, there's real demand for rapid production of food, in particular protein-based food. If we can produce fish, larger fish much more rapidly, we can feed a lot more people, and that can be a good thing. One problem with all of these techniques is that when we're inserting genes into an organism, into a target organism, it's often difficult to determine exactly where that gene is going to be inserted. And that can cause problems. If we insert the gene into the wrong place, we may disrupt a normally functioning gene, and it can lead to problems in the recipient. So if we want to be able to do these things to manipulate organisms, in particular, if we want to try to do this to manipulate humans, we need to be able to direct the insertion of the genes in a way that is not going to cause problems in that host. And one of the most recent discoveries is the discovery of another mechanism that bacteria use to restrict successful invasion by viruses. And this is something called CRISPR. CRISPR is actually a sequence of nucleotides that can bind to a specific sequence of DNA, and it combines with another protein that's called Cas9, which is capable of cutting DNA. And so what bacterial cells use this system to do is to identify viral DNA, and they have a particular sequence that will identify it and bind to it. And then they use Cas9 to cut up the DNA in a way that's very similar to what they did with restriction enzymes. And the benefit of this is that it can be more specific because you've got a specific sequence of nucleotides that will guide the Cas9 to the exact right position in order to cut the DNA. And what researchers have discovered is that you can manipulate the sequence of this CRISPR and attach a CRISPR of any sequence of bases to a Cas9 protein that allows you to direct this complex to any particular spot in the genome that you would like. So you can cut the DNA with Cas9 and insert then sequences of DNA to insert a gene. And this is a technique that is much more precise and much more accurate in inserting DNA into a particular location in an organism. And this is a technology that is just getting started. It's been around for less than 10 years. So the potential for this technology is really unknown. But people suggest that we might be able to use this technique to insert genes to cure genetic diseases, even something like sickle cell disease. So this is a slide that demonstrates how that might work. And it was research that was done on mice that have a genetic disease that's very similar to sickle cell. That is, they're homozygous recessive for a different form of hemoglobin. So what they do is they use these mice that have sickle cell disease, and they isolate certain cells, grow them in culture, and those cells, again, contain the sickle cell genes, and they manipulate them so that they can become stem cells. Stem cells are cells that can then develop into any potential kind of cell. And what we want to be able to do is to get these cells to develop into blood cells and into red blood cells that will have the right kind of hemoglobin. And so these cells can be manipulated using the CRISPR-Cas9 system. And so we can insert into the DNA in these stem cells the DNA sequence for the normal hemoglobin. And that's indicated at the bottom part of this diagram. And once we've done that, we manipulate these cells and grow them in culture and then reintroduce them back into the mouse and hope that these now blood stem cells will replace the stem cells that already exist in that mouse. And these new stem cells are ones that are producing normal hemoglobin. So this is a technique that could possibly be used in order to manipulate human genes in order to treat disease. So there's a tremendous amount of potential in this technique. I think we're years off from being able to do this, but at least this CRISPR-Cas9 system has the potential to do a lot of potentially positive biotechnologies that can be used to treat disease and end suffering. All right, one last slide to describe one last level of technology that you may have heard about, and I think it's worth explaining. And that's creating clones. A cloned organism. Cloned organism is an organism that has the exact same genetic makeup as the source of its cells. And you've probably heard of Dolly the sheep. She was produced using this technique, probably 15 to 20 years ago. And she has since passed away. She died when she was about 6 or 7 years old. But in this technique, what you do is you isolate the source DNA that you want to be cloned. You isolate cells from that source, and you manipulate them in a way that removes the nucleus of those cells, but the cells remain intact and alive. And then you isolate a cell that's going to serve as the source of a nucleus, the source of DNA. So you isolate the DNA of that organism, the nucleus of that organism, inject it into the cell whose nucleus you've removed, and you now have an egg with a nucleus that's come from a different individual. And you manipulate that egg so it can begin to develop, and it develops into the blastocyst stage, which is the stage at which an embryo implants in the uterus. That's anywhere from 1 to 2 weeks after the egg cell starts to develop. And then you insert that into the uterus of a surrogate ewe. And if everything goes right, it will develop into a baby lamb that grows into a sheep. And that sheep's genetic makeup is not at all similar to the mother in whose womb it grew, but it is identical to the individual sheep that supplied the nucleus. So the one on the upper left is actually that sheep's parent. But this Dolly only had one parent. It has the exact same genetic makeup as that original parental cell. These researchers had a bit of a sense of humor because they named her Dolly simply because the cells that they're using as the source of a nucleus were cells from the mammary glands of that mother sheep. And so they named it after Dolly Parton. I know that's kind of silly, but that's why they named her Dolly. This is a technique that has been used. It's worked on other organisms. They've done cats, for instance, and created a clone of cats. And the number of organisms that have been cloned is increasing, but it's still a difficult technique. In the case of Dolly the sheep, it took 270 tries at this technique, and they only had one successful lamb that was born. Most of them, the fertilized egg never started to divide, never started to develop. In some cases, it began to develop, but never developed to the blastocyst stage. It died. In some cases, it did develop to the blastocyst stage, and it was inserted into the uterus of a surrogate ewe, but the embryo didn't survive. So there are all sorts of reasons why that happened. So we have to be really careful with this. It's one thing to do this on sheep, but if we're going to try to do this on humans with the failure rate that we saw, it may not be responsible to do this technique to try to develop one clone when we had 269 failures. So this is a technology that we have to be very careful with, and we have to learn a lot more about to try to understand why the success rate was so low. Once we can figure that out and understand how we can improve the process, then maybe this will be used to clone humans. Who knows? But I think that's way off in the future. But these are techniques of genetic engineering that can be used to move genes from one organism to another to manipulate them in ways that can be beneficial to that organism and beneficial to other organisms. And using bacteria to produce drugs is a very beneficial result of this technique. It produces drugs that are much better in quality, much purer, and are much cheaper to produce than the ways they were produced traditionally. And it also provided mechanisms to produce new and novel drugs that had never been produced before. So individuals with particular diseases can now receive treatment. So I think there is a lot of benefit. There is also, we have to be cautious because anytime we're manipulating the genetic makeup of organisms, there can be unforeseen consequences. So we need to do this very carefully. We need to make sure that we're doing it in an ethical manner. We need to think about carefully the process and the results of this to make sure that we're not hurting any individuals when we do this. So it's something that we need to develop ethical guidelines as we develop the technologies to make sure that we're using this properly. Okay, so that ends the third of our three lectures from our snow day. So make sure you look at all of these, listen to the lectures, look at the PowerPoint slides, and if you have any questions, bring them to our next and last class, and we can go over any of this information that you need to. All right, see you in class.